Well, back in this video, I'm going to talk about DNA packaging, and we'll start off by looking at packaging in prokaryotes, and we'll talk about bacteria. So the general problem is that you've got very long molecules of DNA that must be fit into the cell, in the case of eukaryotes, into the nucleus. So it doesn't matter whether you're prokaryote or eukaryote, DNA packaging is a major problem. It's a major challenge that has to be overcome for all forms of life. And there are different strategies, but some similar and related themes that we can talk about. Let's talk about bacteria to start with. So bacterial chromosomes were once thought to be just naked DNA, just a naked double helix, but we now know that DNA is packaged somewhat similarly to the chromosomes of eukaryotes. The main bacterial genome is encoded on the bacterial chromosome, and then there are extrachromosomal circular DNA structures called plasmids, and we're not going to talk about those. I'll leave that for microbiology. So generally speaking, bacteria have single, multiple, linear or circular chromosomes depending on the species. The most common structure is a single circular chromosome. That's what you're going to find most commonly. And that DNA molecule is bound to small amounts of protein, and it's localized to a region of the bacterial cell called the nucleoid. The bacterial DNA tends to be negatively supercoiled, and it's folded into loops called radial domain loops. Like I mentioned, besides the chromosomal DNA, bacteria contain plasmids, relatively small circular DNA molecules, and they're obviously familiar to you in the context of things like antibiotic resistance, and they don't often code essential functions. These are usually encoding accessory functions, so they can be done away with. They're not essential for survival. So that's a little bit about bacteria. Let's focus the rest of our discussion on eukaryotic cell biology. So in eukaryotes compared to prokaryotes, there is significantly more DNA per cell if you compare genome sizes, and the eukaryotic DNA interacts with many more proteins than you see in respect to bacterial DNA interacting with proteins. When bound to proteins, DNA becomes what we call chromatin, and to separate that chromatin into two new cells at the time of cell division, the chromatin has to condense down into a much more compact structure. Although in an interphase cell, we still have pretty compact structures, which we call the chromosomes, and they become much more condensed and easier to manage during mitosis. The most common form of protein that we're going to talk about here are the histones. This is a group of small basic proteins. They've got a lot of lysine and arginine, which contributes to their basic nature. That means they're positively charged. The negatively charged DNA molecules combine stably to the positively charged proteins, the positively charged histone proteins. And in terms of the amount of histone compared to DNA, if you take the atomic mass of the histones compared to the atomic mass of the DNA, they're about equal. So if you work out the actual mass of DNA in a cell, it's about the same as the mass of histone proteins. That gives you an idea of the fact that both the DNA and the protein is important in DNA packaging. There are 5 main types of these histone proteins: H1, H2A, H2B, H3, and H4. We'll talk about those and the structure they form when we talk about nucleosomes. So chromatin contains about equal numbers of H2A, H2B, H3, and H4. And then there's about half as much H1, histone H1. And then there's a bunch of also of additional proteins called non-histone proteins, which play packaging roles, and we won't really talk about those too much here. So in the 1960s, X-ray diffraction studies, not dissimilar to the ones that Crick and Watson and Rosalind Franklin used to study the structure of DNA, revealed that chromatin has this repeating structural unit that's seen in neither purified histones nor purified DNA alone. And so when you isolate chromatin from cells, you can spool out the chromatin fibers, and they appear as this kind of series of tiny particles attached by these thin filaments, and this is called the beads on a string model. And the beads here are structures called nucleosomes, which you've probably heard about in passing in general biology. So to study the structure of these nucleosomes, is what you can do is you can take chromatin, I isolate the chromatin from a cell, and you can expose it to a nuclease, and that cleaves the DNA. then you can take the partially degraded chromatome, remove the chromatin protein, so you just have left over the DNA fragments. Then when you analyze the DNA fragments, what you find is you have this ladder of DNA of about where each of the steps between each of these bands, which is a bunch of molecules of the same molecular mass, these bands are all multiples of about 200 base pairs. And I've pulled a gel off of the web here so you can actually see what this looks like. And so this would be 200 base pairs difference in size, 200 base pairs, 200 base. And the reason these get compressed up at the top here is because of the way that agarose gel electrophoresis separates DNA. As we're actually looking at here is some cells which have been induced to undergo apoptosis. And so these cells on the left here, 100% of them are apoptotic and 0 are non-apoptotic, all the way down here to 0% apoptotic. So you can see what happens is you get this laddering of DNA when you induce apoptosis in cells, and that's actually due to fragmentation of the nuclear DNA. So this data suggests that these nucleosomes, the actual histone-based structures with DNA wrapped around them that we'll look at in a moment, are separated by 200 base pairs or so of DNA. A pretty famous guy called Roger Kornberg showed that you can actually build these nucleosomes in vitro if you isolate the components pretty carefully. So if you isolate the histones from cells pretty carefully, as what you see is you find dimers of H2A and H2B. That means there's 2 copies of H2A and H2B, and 2 copies of H3 and 2 copies of H4, and you can keep those intact when you purify them. And so they decided that this H2A, H2B, H3, H4 octamer, because there's 2 of each, forms what's called the core nucleosomal particle, and then the H1, there's 1 of those per nucleosome, and that's called the linker histone. And so there's not as many of those as there are the other histones, so you can imagine, you can equally see the ratio of H1 to H2B here. There's 2 H2Bs, there'd be half as many H1s, which is what we said a little while ago. You can also then do an experiment where you isolate the nucleosomal core particle and you treat that particle with a cross-linking agent which binds everything together. And when you do that, is what you find is that you have 146 or 147 base pairs of DNA wrapped around the nucleosome, and then the extra 55 or so, 53, 54 base pairs of DNA is the linker DNA. So it's basically this DNA that's wrapped around the core histone, and the linker DNA is 200, and then we've got the 146 to 147 base pairs, and I say 146 to 147 because it varies from species to species, cell type to cell type, as does the amount of linker DNA, but the core particle is always 146 to 147, so that DNA wraps around 1.7 times. So that's the kind of structure of the nucleosomal core particle and some associated DNA. You can see the diameter of is about 10 nanometers if we consider this as a sphere. So that's the nucleosomal core particle, and the nucleosome is kind of the first step in packaging nuclear DNA. So here's a double helix, naked DNA, and we wind the naked DNA around the nucleosomal core particle of those 8 histones. And then we have the short region of linker DNA, and that gives us our beads on a string model that we looked at just now. If we take a look at the actual chromatin that we find in cells, though, we actually find this what's called a 30 nanometer fiber. So these beads in a string model actually folds up and packages into this 30 nanometers, like zigzagging structure of nucleosomes, to give us this 30 nanometer core fiber, and the association between histone H1s in the different nucleosome core particles is responsible for this level of packaging, and so that gives us this level of packaging. We can then take the 30 nanometer fiber and we can actually create these what are called radial loops or radial domains, and it gives us this approximately 300 nanometer, 0.3 microns loops. They're all different sizes, they're not identical sizes, and we get these loops of 50,000 to 100,000 base pairs, and they're stabilized by a protein called cohesin in animals, and so that gives us this level of structure. And then we can take these structures and loop them up to form this 700 nanometer fiber structure, and then we can condense that even more into what we know as metaphase chromosomes. So multiple levels of packaging. If we're looking at DNA which is being transcribed, then it is less tightly packed than inactive DNA, and that makes sense because a looser packaging conformation would allow easy access to the actual information in the bases, which determines which proteins involved in transcription should bind to specific regions of the DNA. And so we can actually quantify this and put on a packaging ratio which tells us how tightly packed the DNA is. We're not going to worry about that too much. Is what we will just talk about quickly is how in cells you have sections of chromatin that are so highly packed and so highly compacted that they show up as dark spots in transmission electron microscopy. And remember, these darker regions are regions of greater electron density, and so that gives you an idea of the greater amount of matter that's packed into the space here. So this heterochromatin here is very, very tightly packed, and then the euchromatin in the nucleus, this is the nucleus, sorry, it's the nuclear envelope, that's a structure called the nucleolus, and then this light grey molecule, or light grey matter, is less tightly packed chromatin, and that's called euchromatin, and that's going to be metabolically active. There'll be a lot more transcription going on that. Now if we go into mitosis, then all of the chromatin becomes very highly condensed, more condensed than heterochromatin. So there are some types of chromatin which can go back and forth between being heterochromatin and euchromatin, and that's called facultative heterochromatin, literally because it goes back and forth as it needs to. On the other hand, some heterochromatin is permanently compacted. It never ever decondenses and unwinds and becomes more loosely packed. That's called constitutive heterochromatin. It serves several structural functions in chromosomes. Probably the most two important types of structural function here are at the centromere and at the telomere. The centromere is on the left, that's in the middle of the chromosome, and that appears as a constriction of the chromosomal body. It's bound by a complex set of proteins and has numerous functions. For example, it holds the sister chromatids together during mitosis and meiosis before they're separated. It's also the site of the construction of kinetochores, which is where you attach spindle microtubules. If you remember the spindle that forms in mitosis and meiosis, you attach those spindle microtubules to the centromere so you can pull the chromosomes apart. The sequences of DNA at the centromeres there are highly repetitive. They're called CEN sequences, C-E-N, and they are the sequences vary quite significantly from species to species, so we're not going to worry about the sequences too much. Telomeres, these are found at the tips of chromosomes, and they contain highly repetitive DNA sequences, and they're often compared to those little grommets you find on the ends of your laces because they protect the ends of the DNA from being eroded during DNA replication. So today, all chromosomes that have been looked at or vertebrates, have the same telomere sequence, and it's a repeat of the sequence TTA-GGG. Don't remember it, but it is always this 6 nucleotide repeat: TTA-GGG, TTA-GGG, TTA-GGG. So that's what the telomere is composed of. So that's some constitutive heterochromatin. I want to turn our attention now to looking at the genomes of mitochondria and chloroplasts, and your book doesn't do a fantastic job of covering this, so I've added some additional stuff in, so you won't read about this in your book. Hopefully you know that mitochondria and chloroplasts have their own chromosomes, they have their own genomes, they're devoid of histones, and they're usually circular. Both of the chloroplast and the mitochondria genomes encode polypeptides which are used within the organelle, but many proteins that you find working in the mitochondria and the chloroplast are encoded in the nuclear genomes, and that means you've got to think that there are mechanisms to get these proteins into the mitochondria and the chloroplast, because they're not all encoded by the mitochondria and the chloroplast genome. Both genomes have been sequenced. We don't need to talk about the sizes or anything. some nice evidence for the theory of endosymbiosis, which explains, which is one explanation of how we got a eukaryotic cell. So we have an ancestral cell, a primitive nuclear structure, and and this primitive cell will have engulfed a bacteria which was capable of oxidative phosphorylation. So there would have been an endocytotic or a phagocytic event, and so if that bacteria contained its own circular genome, as most bacteria do, then that explains why the mitochondrial genome is still circular. It also explains why the mitochondrial inner membrane, the convoluted one, has the same kind of lipid composition as certain bacterial membranes. And then this cell type here probably then phagocytosed some kind of photosynthetic bacterium, and that led to the evolution of the modern day chloroplast. So that's one explanation of how this occurred. And just like the mitochondrial chloroplast, the mitochondrial genome, the chloroplast genome is also circular. And again, that's probably due to the fact that the chloroplast had its origins in a photosynthetic bacterium with a circular genome. If we go back to thinking a little bit about the proteins that function just in the mitochondria to start with, and we won't worry about the chloroplast, the proteins which are encoded by the mitochondrial genome and versus the nuclear genome, is what we can see is that there are a certain number of proteins. These aren't all the proteins, but these are some of the proteins in the mitochondria. We're just looking at the inner mitochondrial membrane here. I'm not worried about the outer here. There are some proteins which are nuclear encoded, like this protein here, the TIM translocase, and that is a protein which is involved in nuclear import, so for getting proteins into the mitochondria. So all the blue proteins here, those are nuclear encoded, whereas the proteins in orange, those are always encoded by the mitochondrial genome. So you can see there's some, but not all. And then there's the pink ones. Sometimes they're nuclear, sometimes they are mitochondrial genome encoded. Just depends which species, which phylogenetic group you look in. All of these processes up here, all of the proteins involved in these processes are under nuclear control. So these are processes controlled by proteins made by the nuclear genome. So what we got is actually a complicated situation of mitochondrial genomes, which encode some of the proteins the mitochondria needs, but the mitochondria has become dependent on its host cell for certain other protein functions. So when we talk about the mitochondria and the chloroplast being semi-autonomous, this epitomizes why. They can do some things because they can encode those functions within their genome, but their ability to do everything has been lost, and they've developed a dependency on their host cell, which is the cell in which they found themselves many millions, billions of years ago. So that's a little bit about the relationship between the mitochondrial genomes and the nuclear genomes, and we won't talk about the chloroplast, but you see similar kinds of things. Another interesting thing about another interesting difference, I guess, between the mitochondrial central dogma, if you like, and the nuclear central dogma is some variations in the genetic code. And the general biology are told that the genetic code is universal, which means that AGA always codes for the same thing. There's no variation. And I'm afraid that's not entirely true, because there are some exceptions to this. The mitochondria uses slightly different genetic codes, and I remember a class this past semester where a student was working on a transcription translation problem in General Biology 1, and I'd given the solution so that people could see whether they were getting the right answers or not, and she'd said that there was something wrong with the problems because she was getting the wrong amino acids when she did a translation, when she translated a piece of mRNA. And it turns out that the genetic code that she Googled and found was the mitochondrial one, so she actually got some variation. So, I mean, let's just take a quick look at this. UGA is normally a stop codon, so in the standard genetic code, UGA is a stop codon. In the mitochondria of mammals, Drosophila neurospora, which is a type of fungus, and yeasts, another fungus, that UGA actually codes for tryptophan. So there's variation in the genetic code there. Plants, mitochondria, it's a stop codon. So you can just take a look at this, and you can just see that there is significant variation in the genetic code when you look at the mitochondria and compare it to the nuclear, the genetic code that's used to interpret the information in the nuclear genome. And again, that speaks that speaks to some support for the endosymbiotic theory that we just talked about. Last thing I want to finish up on here is something interesting about the mitochondrial and the chloroplast genomes that I don't think you've heard about before. And so this is more kind of on the cell biology spectrum of things than the genetic spectrum of things. There are several mutations in mitochondrial DNA that can cause diseases in humans. And the severity of the disease often depends on the type of mutation and the number of copies that are present in the cells of affected tissues. Now that sounds a little bit strange because you're sitting and you're hopefully thinking, well, what's he talking about? Because humans are diploids, so there's only 2 copies. But when you think about mitochondria, there are hundreds to thousands of mitochondria depending on the cell type. So it's entirely possible that you could have a few copies of a mutant genome and many copies of a normal wild-type genome, or the other way around. And so what you find is that you get a situation in some of these diseases called heteroplasmy. And this is where cells contain mixtures of wild-type and mutant mitochondrial genomes. So how does that happen? When a somatic cell divides, the mitochondria just get randomly divided into the new cells. So anything in the cell which is at a high enough copy number, when you do cytokinesis and you divide that cytoplasm, you just stick roughly half and half. And you do it randomly. There's no way of knowing that this mitochondria has got a functional genome and this one's got a defect which leads to some kind of disease. The cell just says, yeah, let's just split down the middle and mitochondria go into each new cell. So what that means is that the copy number of mutant genomes can drift up or down. So this is really an example of evolutionary biology in action, because as you remember from general biology too, one of the mechanisms of evolution is genetic drift. And so you can have an allele of a gene drift up in frequency and fix at 1, or you can have an allele drift down to 0 and be lost entirely from the population. So the population we're thinking about here is a population of cells. If we are randomly segregating the mitochondria during cytokinesis, we can have a lot of drift up towards 1 or down towards 0, and that's how you can end up with cells which exhibit more severe manifestations of the disease phenotype than other cells. It's because some cells can just by chance accumulate more mutant mitochondrial genomes than others. So there's also some evidence that shows that the accumulation of mutations in the mitochondrial genome is responsible for aging in mammals. And so the experiment that we're looking at here is we've got 2 mouse, a normal mouse, and then we've got a homozygous mutant mouse. And this mouse here, actually, if you look at him physiologically, looks much older. They're the same age, but this mouse here is going to look much older than this mouse here physiologically and physically. So what's being done is this mouse here is unable to proofread the DNA in its mitochondrial. So its mitochondrial DNA polymerase has lost its ability by mutation to proofread errors in DNA. And so the mutation rate in the mitochondrial genome has gone up. And what we see is this mouse ages more rapidly. Now, if we look at that in terms of the survival of the mice, the wild-type mouse, that's this little, the black furred mouse back here, he lives out, 1,000 days. It's 3-year-old. It's not bad for a mouse, and there's a little bit of drop in mortality in the population. So if we took 100 mice, then we'd be looking at maybe, 5 of them will have died by 3 years out here somewhere. So this is just think about this as a population of 100 mice. Wild-type, there's some loss, but not many. Now, heterozygous ones, that means if we've got one defective allele of the mitochondrial DNA polymerase, now we're seeing about maybe 20% mortality out. But if we make the situation homozygous, that means there's 2 defective alleles, or there are, this is nuclear encoded. We mutate both alleles of the DNA polymerase. Now what we see is that 100% of the mice have died by not even a year and a half old. So as the mutations accumulate in the mitochondrial genome, the mice age more rapidly and die sooner. And so we are having cellular effects due to loss of DNA repair fidelity in the mitochondrial genomes. So obviously this doesn't apply to chloroplasts, but what we're seeing here is a loss of lifespan in these mice due to an inability to accurately replicate the genome. So I'll leave you that last interesting piece of information, and we'll wrap it up there. And when we come back, we'll talk about the nucleus. I'll see you shortly.